So if you are not sure on the actual steps of transcription versus DNA replication, or how to talk about those for exam questions, please go back and watch one of the other videos. This is just a laying out of the comparison, because they like to ask about compare and contrast for these two things. But the other thing is that a lot of students get them confused. So it's really important that you guys understand which one you're talking about and using the appropriate vocabulary. Okay, so I'm going to outline some similarities and differences for these two things. So I'm going to talk about the similarities first. Okay, so both of them involve copying the DNA. Okay, and both of them involve free nucleotides. And don't worry, I'll get to the idea that, RNA versus DNA nucleotides, but yeah. Okay, so you're copying the DNA in both of them. You're using free nucleotides in both of them. Excuse me. You could say that they both take place in the nucleus. And the first step of them is both, the idea is that the DNA has to unwind or unzip. All right, but the similarities pretty much end around there. Okay, the differences actually involve a lot of the same ideas. So yes, you're copying the DNA, but I'm going to abbreviate these. So in transcription, okay, you're not, that's confusing. Never mind, I'll just make two. Okay, so I'll do this. All right, so the idea is that while you're copying both of them, you're copying only part, right? A single gene or a small section of the chromosome. Whereas the DNA replication, when you're replicating the chromosome, you're replicating the entire chromosome. So you're copying the entire chromosome or even the entire genome. Okay, so that's one of the big things. And because of that, of course, the end result is different, right? This results in DNA, or sorry, an mRNA is produced. Whereas in DNA, you get a new DNA molecule. Well, a half new DNA molecule technically, but the idea is that it's going from one DNA molecule to two DNA molecules. And of course, in those two, you have one strand of old and one strand of new. But the idea is that it's different, okay? They both involve free nucleotides, but the nucleotides themselves are different, right? So this involves RNA nucleotides. And I would also talk about the different bases so you have A, U, C, and G, whereas this is DNA nucleotides, which are, of course, A, T, C, and G. And now if you wanted to, you could say the similarities are they both have the nitrogenous bases A, C, and G. So you could list that if you want. But of course, the sugars on the nucleotides are different, so they're not exactly parallel, but that idea. Okay, so they both involve A, C, G nitrogenous bases. Okay. They both unwind or unzip, but again, because you're either copying the entire thing or only a single gene, you could talk about not all of the DNA is unwound here. And here it is. So that would be similar to this. I'm not sure that'd be a separate point, but still. All right. So the entire DNA molecule is unwound. Right. Other differences would involve the idea that for transcription, so not only is the mRNA produced versus the DNA, but this is maybe I'll include this up here, a single-stranded is the final result, whereas the DNA molecules are double-stranded. And the other thing that you could talk about is the idea that DNA replication, as you guys know, this is something we've talked about a lot, is semi-conservative. that's the term we use for it. Transcription, you can literally say is not semi-conservative. We don't have an overall term for it, or you guys don't, but it's enough just to say, because this, and it's considered semi-conservative, like they're both producing copies of the DNA, right? But this, because you're only copying a small part of it, it doesn't count as semi-conservative. Whereas this one, it's semi-conservative because of the idea of you have a full new strand and a full old strand. So that's why. So yeah, just the term itself can often be worth a point, and you don't have to necessarily say what transcription is. You can just say it is not semi-conservative. If you wanted, you could also talk about the enzymes that are involved. So right here you have RNA polymerase that is doing the creation of the phosphodiester bonds and unzipping the DNA. Here you have DNA polymerase, or you could mention one of the others, polymerase, ligase, or helicase. All of these are different enzymes. All right, so that's, sorry, there we go. Okay, so there's lots of differences that you can name, obviously, and if they ask about similarities. And again, so if they ask you a compare and contrast question, this is helpful, but also just to get straight in your mind, because these differences are important when you are describing either of the processes, to make sure that you are using the correct vocabulary and language, and you're not drifting into the other process when you're trying to describe only one of them.